Once you've established your model and it's running, you really have this capability of exploring a new biology where we have now shear stress going in, we have the replacement of molecules, we have gradients that are building up inside of the chip, and this is something that traditional cell cultures are just not able to recapitulate. Hello, my name is Adrian Feile. I'm working in the University Hospital in Jena in the group of Alexander Mosig, and the research focus of our group is the study of infectious diseases, and the particular infectious disease I'm interested in is cholerae disease caused by the bacterium Vibrio cholerae. Vibrio cholerae is luckily not an infectious disease in primarily the Western or European population, but it still is of major interest in many other parts of the world, such as Africa or Asia. And the problem when working with Vibrio cholerae is that it is a strictly human pathogen, so it does not really infect different animals or mammals. So using animal models are not really a reliable tool to understand the disease onset. And yeah, that's why we are working here with intestinal-on-chip models. We are working with the Dynamic 42 chip platform. This chip platform has two channels, which is divided by a porous membrane. And on one side of this membrane, we are able to colonize intestinal epithelial cells, which we use KCO2 cells for. And on the other side, we can mimic an endothelial vascular compartment by using HUVEC cells. And we are also including macrophages into the system to give it a more background into the immune system and the response to this infectious disease. And yeah, we have this luminal compartment above the intestinal cells where we are able to then inoculate the bacteria to then study the infectious process of Vibrio cholerae in this model. I can basically start with first the establishment of the model because taking care of infectious models in an organ on chip is actually not as trivial. So we first looked into different time points of the infection up to 12 hours, and we saw, as expected, a natural growth of the bacteria over time. And this growth resulted in a host response where E-caterine and ZA1 as tight junction and adherence junction molecules were starting to disintegrate to this infection of the Vibrio cholerae pathogen. And what was very interesting though to see was that this damage that we found specifically in the tight junction of where we define the ZA1 fragments compared to the ZA1 filaments, which are more the healthy part, that these fragments are much closer linked to where the biofilm is sitting compared to the healthy ZA1. So really, like we see this damage of tight junctions being closely correlated to where the biofilm sits. So this spatial correlation is really quite interesting. On the other hand, this tissue damage then also results in pro-inflammatory cytokine secretion. We have interleukin-1 beta, IL-6 and IL-8, and this probably is linked to our macrophages in the system that elevate this response. And we kind of established this model to then specifically look into quorum sensing of the bacteria, where these bacteria are able to communicate with one another via small molecules. And this is a process that is just quite recently found and which seems to be of high importance in how these bacteria behave and how they organize themselves to respond to different environments, if they're like in the water, in a human or somewhere else, and they seem to coordinate their behavior based on these quorum sensing molecules. And the one particular one we are interested in is DPO. And DPO is a molecule that they can produce by themselves. And previous studies have shown that this DPO signaling via its receptor VQMA responds in the regulation of virulence expression and biofilm formation of these bacteria. So we were wondering if we can understand this pathway more, and if by addition or leaving out of this DPO signaling, that we can manipulate the behavior of the biofilms. And so we did exactly that in our chip, and we co-infected this chip with wild-type bacteria and with bacteria that are deleted in the VQMA receptor, so they are not responsive to DPO. And we could see that only the wild-type bacteria that can sense DPO have a significant outgrowth as a response to this treatment. And we can see that this is not just an overall CFU increase, but we actually have a bacterial increase per biofilm. So each individual biofilm in the end of the infectious period has more bacteria. And this was quite an interesting result, actually, which we were not expecting in this significance. A cool thing with our study is that we are really able to look into these biofilms in a bacterial cell resolution. So really of these biofilms, we can detect each individual bacterium, and we can see its positioning in relation to the intestinal cells. And we can then look into this heterogeneous response that we have in the biofilm. So each dot here basically is a bacterium. And we now looked into VQMR expression, which is dependent on the DPO signaling. And you can see that this expression of VQMR really is very heterogeneous. So some bacteria are responding, some are not responding. And we can see that this response is spatially separated. So it's mostly the apical bacteria in the biofilm responding. And those are also the bacteria that then have this massive outgrowth in their biofilm. So we can see here in the y-axis, the depth of the biofilm. So here we are apical, and down here we are basal. And on the x-axis we have the amount of bacteria per position. And we see that this increase in bacteria that we see with DPO treatment is just happening in the apical part of the biofilm, whereas in the more basal part there's no difference. So we really see the spatial differentiation inside of the biofilm that we can create with the quorum sensing manipulation. So this is really cool, and it shows how this quorum sensing really seems to have a major impact on how the bacteria behave in the chip. And hopefully, this gives us more insights in how the bacteria are responding and living inside us humans, but also inside of the environment. When you start with organ on chip, you're first bombarded with all these technicalities, with tubings, with different ports and molecules. So I would recommend to really start simple. Don't start with the biology, just start with adding water to the chip, add all your different components like tubings and so on, and figure out how you can work bubble-free and how you can use the technology in the correct way. And once you have established that, then you can start into going more deeper into the biology. And the cool thing then is once you've established your model and it's running, you really have this capability of exploring a new biology where we have now shear stress going in, we have the replacement of molecules, we have gradients that are building up inside of the chip, and this is something that traditional cell cultures are just not able to recapitulate.